The overall goal of this procedure is to identify and locate major translational pause sites for an mRNA translated in a cell-free translation system. This is accomplished by first cloning the gene of interest under the control of a T7 transcriptional promoter. Next, mRNA is transcribed and purified. Then, in vitro translation is performed in the presence of mRNA and radioactive amino acids to label the synthesized protein and nascent polypeptides. Finally, the polysomes are isolated using sedimentation velocity centrifugation, and the isolated nascent chains are resolved by gel electrophoresis. Ultimately, results can be obtained that show nascent polypeptides of different lengths by tris-trisine SDS page and autoradiography, thus indicating the presence of the pause sites along the mRNA of interest.
The main advantage of this technique over existing methods like micrococcal nuclease protection assay is that it's extremely simple, fast, and efficient, and allows determination and identification of the major translation pause sites along any mRNA in a given cell-free system. This method can help answer some of the key questions in the field of translation and co-translational protein folding, such as how factors like codon usage, tRNA abundance, and/or mRNA secondary structure may affect kinetics of translation. This method may further help our understanding of the relationships between the kinetics of translation and the key steps in the co-translational protein folding pathway.
The implication of this technique extends towards understanding the process of protein folding in vivo, which is largely